Hello everyone, and thank you for attending our symposium. In this talk, I am going to show you some of my recent advances in the integration of physiology and behavior for the estimation of climatic vulnerability in tropical lizards. As a rule of thumb, when an ectothermic animal such as an insect, a frog, or a lizard performs a function, it will do it increasingly better as its body temperature rises from a critical thermal minimum temperature to an optimum temperature that maximizes the organism's performance. After that, at higher temperatures, the performance will quickly decrease down to a critical thermal maximum that will block again the function and might rapidly kill the animal. For that reason, ectothermic animals exhibit voluntary thermal maxima and minima that keep their body temperatures within adequate thermal ranges. This is useful because one can compare now measures of thermal tolerance, like this curve, with the body temperatures measured for an animal or estimated for the future as a result of global warming. In that way, one can tell whether a species is or will be exposed to unsuitable temperatures. From doing this calculation for species of different latitudes, the pattern arising is that tropical species might be at higher climatic risk than species of higher latitudes. Now, when ectothermic organisms are acutely exposed to increasingly higher temperature, their probability of survival also decreases exponentially. In addition, this probability of survival might decrease too when they are exposed to lower temperatures but for longer time. As a result, locally, populations of ectotherms track microhabitats with suitable temperatures. In this example, we studied the distribution of environmental temperatures at a gradient from the semi-deciduous forest to an adjacent scrub savanna in the only known region occupied by this little endemic lizard of northeastern Brazil. There, we found that the only places occupied by these lizards were the ones that very rarely exceeded their voluntary thermal maximum, even though our sample sites will be sometimes a few meters away from each other. Now, this reasoning at larger geographic scales, let's say across the whole Amazon basin. With the proper tools, we can also estimate the number of sites where available temperatures will rise over the thermal tolerance of an organism, for how much time along the year this will happen, or the proportion of a species known locations that are exposed to too hot temperatures. Cool, right? There are complicating factors, and one is water availability. In this meta-analysis of 1,900 species of animals and plants, Johanna Brady and Brian Leong in 2016 found that water availability, either in form of precipitation or distance to water, are as important or more than temperature in driving species geographic distribution. How do they interact with each other in tropical regions? If we think of a tropical rainforest, we can visualize that in addition to the strong sun radiation able to cook any organism, there is also so many clouds and also vegetation cover, and also a lot of water available in form of precipitation, watersheds, and a lot of evaporated water. The presence of all humidity has a lot of thermal inertia. The thermal oscillations of the oscillations of air temperatures within narrow thermal ranges. In drier tropical environments, such as a savanna, even though vegetation can still provide shade, the lesser amount of water available means that sun radiation is invested into raising environmental temperatures instead of evaporating water. To understand all that, my students, Carolina Molina Guevara, Cleverson de Sousa Lima, are interested in developing systems to measure the thermal tolerance of different types of organisms and understand how they vary due to dehydration and other factors. The voluntary thermal maximum is a faster and more ethical measure of thermal tolerance for animals, and so far it seems thermal tolerance decreases with dehydration and the voluntary thermal tolerance too, meaning that animals become more thermally conservative as they become dehydrated. Now, the new results I'm going to show you try to answer the following question. Does hydration level affect lizards' VTMAX and vulnerability patterns within the Amazon basin? I joined a zoological inventory across the Rio Negro in northern Brazil. I studied the voluntary thermal maximum of Loxopholis percarinatum, a very small lizard with 1 gram in total weight that lives in one of the most water buffered habitats among terrestrial ecosystems. The voluntary thermal maximum is very simple. One puts the animal to rest in a chamber where it feels safe and will remain quiet. Then one heats it up homogeneously until the animal leaves the chamber. When that happens, one can record the body temperature using a thermal camera. Because these measures vary from individual to individual, I measure the VTMAX of each lizard for 3 times. One in 30 hours of collection, then again after dehydrating the animal overnight, and then I will remeasure it after a process of rehydration of 24 hours. I did that for 19 specimens, and I would always wake the animals right before the measurement of the VTMAX to know if the treatments actually work. Fortunately, they did. Our treatments were able to dehydrate and rehydrate the lizards, and what we found is that the VTMAX would increase with hydration level as expected. Now, to see if the voluntary thermal maximum will be exceeded by environmental temperatures across the Amazon, we use one of the most sophisticated biophysical modeling algorithms out there. The R-niche mapper, implemented in R language by Michael Kearney and Warren Porter in 2016, is able to use heat and water transfer equations to estimate year-round body temperatures for a body of a given size and shape at a given location. I repeated these calculations for thousands of locations within the Amazon basin. Now, this algorithm uses many parameters, and thus its outcome is going to vary according to any of them. Because of that, to evaluate the relative importance of dehydration on the resultant patterns of expected vulnerability, what I did was to create maps of vulnerability comparing all possible combinations of 6 factors, each one with 2 levels. These were the change in body weight related to the dehydration process, the change in VTMAX due to the dehydration process, a change in halving the current rainfall, the current amount of rainfall, and a total decrease in the water surface available for evaporation. I will change also the tree cover from 100% to 80-100%, which is among the is around the range that is observed for these lizards, for these Loxopholis. And I also would allow the animal to bury down to 10 centimeters below the leaf litter. By making all these factors to vary, I generated a lot of models of the temperature of vulnerability at which these lizards will be exposed to different levels of temperature. In that way, I could compare the changes induced by each of these factors. From factors, what I found is that the voluntary thermal maximum, the amount of tree cover, and the amount of rainfall will be the most important factors. Between them, the changes in voluntary thermal thermal maximum would have an order of magnitude more importance for the 3 measures of vulnerability that I used: the amount of locations with exceedingly high temperatures, the amount of yearly time with exceedingly high temperatures, and the amount and the proportion of populations with exceedingly high temperature. The take-home messages now are that, or resultant from this study, are that dehydration alters the thermal tolerance and the geography of expected vulnerability, that knowing geographic patterns in climatic vulnerability requires hard-to-get physiological data, and that changes in tree cover and rainfall need to be watched in the Amazon. And that is all for now. Thank you very much. I want to thank especially to Miguel Trefort for being the coordinator of the faunal inventory, to Liana Brunes, with whom I had the pleasure to prepare and execute the project, and the herpetology lab group from the Biosciences Institute of Sao Paulo University. I want to thank also the Sao Paulo Research Foundation for paying the study, the CAPES and Marie Curie program for paying my bills at the time of the study and now respectively. Thank you to all very much, and see you in the discussion section. Bye bye. Good afternoon.